Yes, dear audience, GS Paul again. We started with meiosis, if you remember, and I have finished meiosis 1. I told you that meiosis takes place in two stages, meiosis 1 and meiosis 2. We have done meiosis 1. Now, before I start with meiosis 2, I would like to review what we have done in meiosis 1 so that you can better understand. Now, in meiosis 1, if you remember, before meiosis 1, there is an interphase, and during that interphase, the cell undergoes the chromosomes of the cell undergo replication. That is an interphase. So you have to remember that chromosomes have already replicated, duplicated, and the meiosis 1 starts. Now, I'll start with meiosis 1. Now, in meiosis 1, there is a prophase 1, metaphase 1, anaphase 1, and telophase 1. If you remember, in prophase 1, the important thing that happened was crossing over. The genetic material from sister chromatids was transferred to the other sister chromatid. Now, in metaphase, the chromosomes line up, and in lining up, they lined up the assorted independent assortment. You have to remember this, independent assortment. That was in metaphase. And in anaphase, if you remember, the chromosomes were separated. And in anaphase, when the chromosomes were separated, the diploid chromosomes changed into haploid chromosomes. Anaphase. In telophase, the cell divided in the middle, and we got two daughter cells with haploid chromosome, and before this, the two daughter cells were formed. They undergo meiosis. They undergo a stage called interkinesis. During this interkinesis, before metaphase, before meiosis 2, in this interkinesis, the cell undergoes a period of rest and grows. Now, during this growth period, a complete cell is formed. A complete cell is formed with the chromosomes, with the nuclear membrane, the nucleus, the centrosome, and the Golgi body, and other cell organelles. In some cases, it is possible that chromosomes do not undergo the formation of chromatin. They straight away grow like the replicated chromosomes. But the nuclear membrane will appear. So, in interkinesis, what happens is the centrosome, single centrosome, the nuclear membrane appears, the nucleus appears, and the chromosomes might change into chromatin material. But there is no replication, there is no duplication of the chromosomes. So the daughter cells is now ready. There are two daughter cells formed. It's ready to undergo Meiosis II. In Meiosis II, I will start. This first thing is Meiosis II also takes place in similar steps: prophase 2, metaphase 2, anaphase 2, and telophase 2. In prophase 2, the nuclear membrane, if it is formed, mind you, if it is formed, it disappears, and the centrioles divides into two, and the two centrioles, they move towards the poles, and they developed the microtubules, and the aster apparatus, so-called, is developed. And the duplicated or replicated chromosomes are thrown into the aster apparatus. And you have to remember that each chromosome has a chromomere, and this is at the chromomere that the microtubules will attach itself, but that is later on. Now, this is what happens in the prophase 2. In metaphase 2, the chromosomes line up in the middle, and of course the sister chromosomes line up in the middle. They are already replicated. Therefore, I'm calling them sister chromosomes. And then they get attached with these microtubules to the centrioles at the poles. So, in metaphase, the chromosomes, the sister chromosomes, I will say, they are lined up in the middle. And in anaphase, the microtubules contract and the chromosomes are pulled towards the respective poles in such a way that the chromosomes come close to the centriole. By the way, at this stage, a constriction might start appearing in the cell. This is what anaphase. The chromosomes have now separated. This is the chromosomes. And in telophase, the constriction deepens and the two cells are formed separately. Two daughter cells are formed separately, and these two daughter cells have haploid chromosome, that is, the number of chromosomes is reduced to N. The nuclear membrane makes its appearance, and the centriole can also be seen, and the other cell organelles also make their appearance. I mean, the Golgi body, the mitochondria, the nucleolus, etc. But these two daughter cells with haploid chromosome and a number of chromosomes are now ready. Consequently, if you remember, after meiosis I, there were two daughter cells. And now, at the end of the meiosis II, there will be four daughter cells. And each four daughter cell have haploid chromosomes. And if you remember, this process of meiosis was brought in for the process of sexual reproduction. So these are the precursors. These are the cells which will form the sexual reproductive structures, sexual reproductive units. I mean, the gametocytes, which will be, in case of males, these gametocytes are spermatocytes, and in females, these will be oocytes. Now, we will see how these ones change. The four cells which are formed change into gametes, I mean sperms and ova. which will mean spermatogenesis and oogenesis. We shall see spermatogenesis and oogenesis separately. Now that is meiosis II. And meiosis I I have already said. And I have already explained this is the process of reductional cell division has been brought in for sexual reproduction. So we shall see how the sexual reproductive gametes, I mean the spermatocytes and oocytes change into the sperms and the ovum. That's all in meiosis II. And as I always say, I'm sure if you like it, thumbs up. And I'm sure you should like it. This is what is happening in you. This is not happening in somewhere else. I mean, you means both in men and women. And so you should like it. And so thumbs up. And if you have any question, I'm sure you'll have some questions. Please write those questions in the comments section. I will answer those questions as soon as possible. And that's the end for today. Thank you very much.